morning, everyone. This morning I'd like to tell you about some work I've done recently in looking at alternative splicing in single cells. So first, by way of background, why are we interested in looking at RNA from single cells? The short answer is that single cell sequencing is one of the only ways to access within tissue heterogeneity. So you can think of this as trying to study the properties of strawberries and bananas using a strawberry-banana smoothie. And that's analogous to what people do when they try to perform bulk sequencing on a heterogeneous mixture of cells. So if you do that sort of experiment and try to make any conclusions about the constituent components of the mixture of cells that you're assaying, you may come to some incorrect conclusions about what makes up the mixture. So single-cell transcriptomics allows us to study transcriptome dynamics across a heterogeneous population of cells. And using this sort of assay, we can identify cell types within a heterogeneous mixture, and we can also study how cells change during a sequential process. So my talk is titled Robust Detection of Alternative Splicing in a Population of Single Cells. So let me first explain alternative splicing and what it means to have alternative splicing in a population of cells. So alternative splicing is just alternate usage of exons that are transcribed from a gene. And there are several ways that this can happen. You can have exon skipping, alternative 3' or 5' splice sites, mutually exclusive exons, or intron retention. Now, when we talk about alternative splicing, what we're really interested in is slightly different from the idea of differential gene expression. So with differential gene expression, which is sort of the traditional way people look at single-cell RNA-seq data, you're interested in identifying differences in absolute or relative expression level of genes between cells. So the number of transcripts that are present, the total number of transcripts, is important. With alternative splicing, we're interested not so much in the total number of transcripts, but in the ratio between the isoforms of an alternatively spliced gene. So for example, if we compare cell A and cell C, they both have a 2-to-1 ratio of isoform 1 and 2, even though the total number of transcripts is different. So that would not be an alternative splicing change. What we're interested in is cases where the ratio between the isoforms is different. Now with single cell data, one type of question that you can ask that you can't really ask as easily from bulk data is what I sort of like to call a single group differential gene expression analysis, where instead of looking at group A versus group B, you're looking at one population and asking what isoform ratios vary within that single population. So that's what we're interested in doing in this study. Another important consideration here is the technical noise that is present in single-cell RNA-seq data. And what I mean by technical noise is that if you do an experiment where you take a single cell volume of identical RNA and split it into, you take a bunch of RNA and split it into single cell volumes and then sequence the individual volumes, you would hope that if the experiment were perfect, you would get identical results from sequencing those single cell volumes. But what you actually see in reality, and this is a figure from Brennecke Nature Methods paper, what you actually see is that rather than lying along the y=x line like you would expect for a perfect experiment, the single cell data have deviations from that line, and those deviations represent technical variation in the measurement. And there are two important properties of this technical noise profile that Brennecke identified. One is that the effect worsens with decreasing volume. So if you start out with a volume of RNA corresponding to a bulk experiment, the noise profile is not very bad. But as you dilute to smaller and smaller volumes, you get increasing amounts of noise. The other effect is that the noise decreases with expression level. So as you have a higher expression level, the noise observed for that gene will be lower. So we developed a method called single splice to identify alternative splicing in single cells. And the method has 3 steps, and I'll go through each of these in detail. But by way of overview, we first identify regions of genes that can potentially undergo alternative splicing. And then for each of the regions, we fit a noise distribution that describes how we expect it to vary based on technical noise. And then we estimate the variance that we expect to see for the ratio of isoforms using bootstrapping. So first, detection of portions of genes that can undergo alternative splicing. So to do this, we construct a splice graph directly from the aligned RNA-seq reads. And a splice graph is a graph in which vertices represent transcribed regions and edges represent splicing events. So we take this graph and decompose it into alternative splicing modules. And an alternative splicing module is a portion of the graph that is single entry, single exit, and each path through an ASM has the important property that it's uniquely identifiable by at least one kind of spliced read. So we can completely identify each path by at least one kind of unique read. The ASMs also have the important property that any alternative splicing event will cause a change in the ratio of at least one pair of ASM paths. Now, one of the reasons why we're doing this whole exercise is that with short read RNA-seq data, as you probably know, it's often impossible to completely resolve a full-length transcript if you have exons that are longer than the read length. So this allows us to zoom in on portions of the gene where we can be confident of the different ratios between the alternative splicing events. Okay, so the second step of single splice is once we have ASMs, we fit technical noise distributions to describe how we expect each ASM path to vary. And to do this, we model the technical variation in terms of two components. One is with probability P, you get a zero measurement, a dropout event. And the other is with probability 1 minus P, you get a sample from a gamma distribution. And going back to the observations that I made earlier about how technical noise varies with expression level, we expect that we can parameterize this distribution by the mean expression level of an ASM path across the set of cells. So in order to do this, we need to learn the parameters P and K and theta that describe the way that genes vary across the set of cells. So to do this, we use gamma generalized linear model regression to estimate variance and logistic regression to estimate the dropout probability. And then from those, we can compute the parameters of the gamma distribution. So after we have noise distributions that describe the way we expect ASM paths to vary, the next step is to compute an empirical distribution that describes the way we expect ratio variance to behave. So to do this, we take a pair of ASM paths A and B and sample from the noise distributions that we use to describe the behavior of both of these paths. Then for each of the samples, AI and BI, we compute the ratio, AI over AI plus BI, and compute the sample variance across the set of cells for that ratio. This gives an empirical distribution on the variance of the ratio across the set of cells. And we can then look at where the observed variation in the ratio falls and compute an empirical p-value for the hypothesis that the variation contains biological noise as well as technical noise. All right, so that's the single splice method. So we next evaluated our method using spike-in transcripts. And spike-in transcripts are exogenous RNA that's added to the cells before reverse transcription and library preparation. And the key property of the spike-ins is that they're added in constant known amounts across the set of cells. So using spike-in transcripts, we constructed two kinds of test sets. One kind is true negatives, and the other is true positives. So in the true negative test set, what we did was we took pairs of spike-in transcripts and treated them as if they were isoforms of an alternatively spliced gene. Now, since the spike-ins are added at constant amounts across the set of cells, any variation in the ratio between a pair of spike-in transcripts should come exclusively from technical noise. So in this case, if we ask single splice whether this pair of transcripts shows alternative splicing or variation in ratio, it should say no. To construct a set of true positive test cases, we took, again, a pair of spike-in transcripts, but in this case, we swapped half of the measured expression levels across the set of cells. So in this case, we have technical noise and simulated isoform switching. And when we ran single splice on all pairs of spike-in transcripts constructed in this way, we found 85% specificity and 86% sensitivity. Okay, so we then applied single splice to real data from mouse embryonic stem cells. And the data set we used was from Buettner paper, and we found 797 genes that show significant alternative splicing across the set of cells. Now, the transcriptional differences among these ES cells primarily come from two sources. One is just biological noise, stochasticity, and the other is cell cycle stage. So to investigate these 797 genes in more detail, we then looked at whether there was any relation between the alternative splicing events we found and cell cycle stage. And the particular data set that we used was nice because before the cells were sequenced, they were experimentally cell cycle staged. So for each cell, we know which cell cycle stage it was in. So we then took that cell cycle information and did a Kruskal-Wallis nonparametric ANOVA test and found 124 genes that show alternative splicing that's linked to cell cycle stage. So now we have genes where alternative splicing events are changing according to the cell cycle. And when we took the isoform ratios for these genes and did PCA, we were able to grossly recover the cell cycle stage of the individual cells using only isoform ratios. And I'm showing here three interesting examples of the genes that we found. HNR-NPC is a ribonucleoprotein, and there's an alternative exon boundary event where the longer exon form is mainly found in S phase, and the shorter exon form is the predominant form in G1 and G2M phases. We also found the SNHG3 gene, which is a relatively uncharacterized long non-coding RNA of unknown function, and it shows a steady decrease in the amount of this top splice variant as you move from G1 to S phase to G2M phase. We also found the RBM25 gene, which is an RNA binding protein, and the distribution of this ratio of these two splice variants across the set of cells is intriguing because it's a bimodal distribution. And what that means is that in most cells, either this splice variant or this splice variant is present, but not both. And you can see that the bottom splice variant here is the predominant variant in G2M phase. Okay, so now I'm going to tell you a little bit about the most recent application of single splice that's still ongoing. We're investigating alternative splicing in the neural cortex. And to do this, we're using data from Darmanis' PNAS paper 2015. And in this paper, they looked at 466 adult and fetal cells from the human neural cortex. And they found a bunch of different cell types, which are shown here in this plot. When we ran single splice on this data set, we found a total of 985 genes that show alternative splicing across the set of cells. So the next question then was whether the alternative splicing events that we found related to any of the cell types that were discovered in this study. So the first thing that we investigated was the difference among fetal quiescent, fetal replicating, and adult neural cells. So again, we used a Kruskal-Wallis test, and one of the genes that came out at the top of the list was SCN2A. And when I looked up this gene, it's sodium voltage gated channel alpha subunit 2. And you can tell that this exon is used much more in adults, and this exon is used much more in fetal tissue. And when I looked up this gene, I found a paper from 2010 where they discovered that this exact splicing event that single splice pulled out is an alternative splicing event in which this SCN2A gene switches from a 5N exon to a 5A exon, and that's the very splicing event that single splice pulled out. Another gene that came out at the top of the list is nucleophosmin, NPM1. And in this case, we have an exon skipping event. And the exon inclusion event is more common in adults than in fetal tissue. But one thing that you can see here that's really interesting is that all of the replicating cells, replicating fetal cells, almost exclusively are transcribing the exon skipped version. And when I looked up NPM1, this is a gene that's known to be involved in neural stem cell proliferation. So it could be that this exon skipping event is allowing these cells to replicate. And with that, I'll conclude and thank my funding sources, and thank you for your attention.